lecture 2 of physiology. And I am Vishwanath Tripathi from FMCH Baleswar. And today we are into the second lecture of general physiology. So we have started general physiology. First lecture is over. So you can go and check out the lecture 1 from my list of videos where I had taught about just giving you an overview of the previous class. We talked about the internal environment, about the homeostatic mechanisms, the positive feedback and the negative feedback. We talked about the gain of the feedback systems. Okay, how to calculate the gain and what is the effect of signs, the plus and minus sign in gain. We talked about that. We compared the systems based on the gains. We talked about positive feedback mechanisms in detail. Negative feedbacks are maximally present, so we don't need to talk about that. So basically, we talked about positive feedback, which are few in numbers as compared to negative feedbacks. Okay, so we talked about positive feedback and mnemonic scar. We talked about shock. We discussed this diagram from Guyton and about LH search, oxytocin. I told you parturition. Uh, at last, we talked about the feed forward and the adaptive control. Right. So this was about your lecture 1, and in today's lecture, 12th April. So in today's lecture, we are gonna cover a very important part. After discussing this, we'll cover the cell membrane. Okay, so cell membrane is commonly asked as a long question even in your physiology exam because it is a scoring part. Uh, if you talk about cell membrane, you read about cell membrane in NCERT. Okay, you read already, right? And you might have read cell membrane in your biochemistry. If you take the Lippincott book, then cell membrane is given in detail and the cell chapter cytology part, right? And today we'll talk about cell membrane basically, and let's see how much we can go. If cell membrane is done, we'll start the intercellular junctions, or if not, then the intercellular junctions will be dealt in the next part, right? So let's talk about cell membrane today. You know many things about the cell membrane. Like it's the outermost covering of a cell. Okay, you must have observed the cell membrane. You must have drawn the cell membrane. A diagram of cell membrane is important. So let me bring a basic diagram of cell membrane. We'll discuss the diagram. Then afterwards, we'll start discussing what are the components. Right. So let me bring a diagram first. So this is the basic diagram of the cell membrane which I got from the internet, and you can see the cell membrane is basically a lipid bilayer. You know all these things. We have an outer face and an inner face, right? And you can see these are the lipid molecules which are arranged like this. These are the hydrophilic heads and the hydrophobic tails. We'll discuss all these things. Just I'm giving an overview of cell membrane, right? And you can draw this diagram. So I will draw a simplified diagram for you even. So don't worry, just see the things first. Then we have the sugar side chains. Here the sugar side chain is attached to the protein, so we call it as glycoprotein. And in case the sugar side chain is attached to the lipid molecule, this sugar side chain we call it glycolipid, right? So we have glycoproteins and glycolipids as well. Then we have cholesterol. Cholesterol is basically a membrane stabilizer. We call it as a fluidity buffer. Fluidity buffer means it maintains, or you can say it helps in the liquefaction or solidification of the cell membrane as per the conditions, or it may act as a membrane stabilizer. Okay, so it is a membrane stabilizer. Then we have the integral proteins or intrinsic proteins. Intrinsic proteins are the proteins which span the total cell membrane, right? And we have then peripheral proteins or extrinsic proteins. These are the proteins which lie outside or inside the cell membrane, right? They may be peripheral in position. They don't span the total width of cell membrane, right? And phospholipid, you can see here, phospholipid is there, and a fatty acid tail of phospholipid. So this is a phospholipid bilayer. So we have one layer of phospholipid here, okay, and two layers basically of phospholipid. So it's a phospholipid bilayer, right? You know all the things about cell membrane, I believe. So I don't need to talk much about that. Okay. If I want, yeah, in today's class, we'll also discuss the transport across cell membrane because that's the main important point which you have to discuss. Transport is a bit in detail, we'll talk about that. But these parts, which are a basic overview of cell membrane, I'll give you an overview. Okay, what I'm teaching is enough. Okay, you don't need to like mug up any other things which are given. If possible, I'll show you some important points from the Ganong textbook as well, if you want for extra edge, right? Okay. So, first point, just write in cell membrane. As basically this is a comprehensive course on physiology, so I'm writing all the points. Just move with my pace and will ace physiology like anything, right? So, cell membrane, the basic thickness of cell membrane is 7.5 nanometers. Okay, 7.5 nanometers. So, thickness is 7.5 nanometers, or you can write 75 Armstrong. Okay, the thickness is basically 75 Armstrong of the cell membrane. This is a fact. And cell membrane has a lipid bilayer with hydrophobic heads and hydrophilic heads and hydrophobic tails. Okay, so it is a phospholipid bilayer. Let's use a symbol with hydrophilic, that is water loving head, and hydrophobic tails, right? This is the whole fact. So basically, you must have seen the basic structure. It's like that. This is a basic carbon chain. Okay, and again, okay, there is a kink in the carbon chain. Okay, there is a kink here. Kink means a fold here. This fold comes due to unsaturation. Due to unsaturation, it comes due to unsaturation. This is the head part, and this is the tail part, right? And this head part is water loving. So this is hydrophilic, and the steel part lies within the membrane, so it is hydrophobic, right? If you know these things, then it's well and good. Okay, so we have hydrophobic heads and hydrophilic tails. You know these things. And what are the types of proteins here? So proteins, let's draw a cell membrane. If you want, you can draw this diagram with me. As it is a comprehensive course, I'm trying to cover each and everything which are there. You know about the discoveries and all. I don't need to repeat that. It is the Singer and Nicholson model, most accepted. Your Singer Nicholson model, given in 1972, right? If I'm not wrong. Yeah, yeah. So this is the thing, and let's draw a intrinsic protein here. This is a intrinsic protein. This is a peripheral protein. Okay, you can call this as intrinsic membrane protein and a peripheral membrane protein. Okay, so what are the functions of the proteins? Proteins have a varied roles. Okay, so roles of proteins. Proteins have widespread functions. Proteins have widespread functions. That is, many functions are there. Like some proteins act as receptors. Okay, receptors. So majority of receptors for body are the proteins itself. They act as antigens, like your ABO system, A antigen, B antigen, you know, right? And they act as transport proteins. They act as carrier proteins, in fact. Okay, for transport. Okay, they are helpful for transport. Some proteins are like forming channel type of things. Okay, if I want to draw, then you know about the ion channels, right? So these are kind of forming the pores. So this is a pore, and this porous type of proteins in which there is a hole in between, right? These are called your channel proteins for movement of substances and ions, right? So these are channel proteins. Channels for movement of ions and all, right? And some proteins act as enzymes, very important. Some proteins act as enzymes. So majority, almost all enzymes I can say are proteins except your ribozyme, which is an RNA enzyme. Okay, ribonuclear enzyme, ribozyme, right? So these are the things which are present. This work of proteins and all. Okay, and these are the tails. Let's draw the tails fast. Okay, so lipid heads and the tails of lipids. Done. Okay. Then in between we have some things like this, some chains. These are called your, here let's make some here. Okay, this one. This one is a cholesterol. Cholesterol. Okay, cholesterol. Cholesterol acts as a fluidity buffer. Fluidity buffer. Fluidity buffer means it maintains the fluidity of the membrane. Where there is a need that membrane must be a bit more hard. Okay, it maintains the hardness or it maintains the softness of the membrane, basically altering the fluid balance across the membrane. So it acts as a fluidity buffer, right? And cholesterol is a membrane stabilizer. So this is basically stabilizing the membrane, right? So cholesterol is there, and any other thing left? Okay, these are the things, and we have many types of proteins as well. We have some alpha helix proteins as well, like some spiral proteins are there. If I want to draw, let me draw. Some proteins are like this. These are called alpha helix protein. Okay, alpha helix proteins. These things are there. Channel proteins and peripheral proteins. I talked about that. Integral proteins. Yeah, lipids I left out. So those lipids. Let me draw the lipids in pink. Not the lipids, carbohydrates. Sorry. Those carbohydrates which are linked to the proteins are known as glycoproteins, and those which are linked to the let's say here we link it here like this. So these are called as your glycolipids. Did I? Yeah. So these are the lipids now. So glycolipids, and here we have glycoproteins. What I'm writing, I don't know. Yeah. These things you understood, right? So that was about your basic understanding of the cell membrane. So basically, if you write this much in cell membrane, if there is a long question, I'm telling about the max which it can come is a long question. Any other question is unlikely from the cell membrane. It won't ask, someone won't ask about the short note about cholesterol. Or I can teach you a lot about a lot things about cholesterol. 27 carbons are there. This double bond is present. This both carbons, and how many chains are there? Okay, CPP rings and all. Many things I can teach you, but as per a physiology point of view, I'm covering that much things which are apt for your exam point of view, right? So about the basic introduction of cell membrane, you have to write this much if it comes as a long question. So in introduction, we'll write these things, right? In introduction, you wrote these things. Let me move to the next part, that is transport across the cell membrane. Okay, transport across the cell membrane. And transport across the cell membrane is a very, very important long question. Either it may come as a long question associated with your cell membrane, or it may come as a separate long question. So let's move to the very important part, that is your transport. Transport across cell membrane. I'm telling you once more, this is a very, very important long question, or it may come as a combination question with your cell membrane. Okay. So transport, basically we have two types of transport. One is the passive transport, one is the active transport. Okay. So we have a passive mode of transport, we have a active mode of transport. You all know this. Passive transport means down the concentration gradient, okay, downhill. Active transport means uphill process. Passive transport, down the concentration gradient you are coming, like if you are running down the stairs, then the gravity is helping you, right? So you are coming along the gradient, right? So it is along the gradient. And this active transport is against the gradient. You are moving up the stairs, right, against the gradient. This if you are moving passively, you have no energy requirement, no ATP needed. Just a minute. Sorry for interruption. There was a call. Okay. So passive, I was telling that it will move down along the gradient. So along the gradient means there will be no ATP requirement, no ATP required. And here there will be ATP requirement. Yeah. Passive transport may be divided into four types, basically four types. Some books may classify into two types, some book may classify into three types. Okay. So basically, one or other make, you can say, like overlap with each other, but let me classify it into four types based on the things which are there. Okay. So here we have four types, and four in first one is a simple diffusion, which is a passive type of diffusion. Simple diffusion. Second one is called as a facilitated diffusion. Third one is called as your non-ionic diffusion. Important, non-ionic diffusion. You can classify this non-ionic diffusion into any of the simple or facilitated types. So it is a overlapping condition. And fourth one is the osmosis. Majority of the books you will see write only 1, 2 and 4. This non-ionic diffusion is sometimes not mentioned in that part. I will tell you what are the things and how you may classify it, and, and other examples of non-ionic diffusion. We have three examples of non-ionic diffusion. We'll remember this at that time when I will teach you one by one, right? And active transport are of two types. You might have knowing this. There's a primary active transport. There's a primary active transport, or it may be a, this is not treatment, this is transport. Okay, don't get confused, right? Primary and secondary active transport. So primary active transport, I will teach you the sodium potassium pump, for example. And secondary active transport, you may have two types. One may be or you can say three types. You can classify the uniport in facilitated as well. So you can say two types. You just remember for understanding. One is import, one is antiport, right? The secondary active may be of two types again. One is antiport. Antiport means the molecules are moving in opposite direction, and another one is symport. Symport means the molecules are moving in the same direction, right? So symport and antiport. And these are the things which are important as per your transport across cell membrane is concerned. Okay. After this, let's discuss the passive transport, that is a simple diffusion first. We'll start with the passive transport. We'll start with the passive transport. And in passive transport, we'll first discussing the simple diffusion. We'll discuss the simple diffusion first, right? Simple diffusion. We'll write the salient points as well. Simple diffusion, what are the substances which are transported by simple diffusion? Basically, you understand that you might have a idea of respiratory physiology, right? That O2 exchange and all. So, exchange of gases is the major thing which is done by the simple diffusion. So, exchange of gases occur by this. Exchange of gases will occur by this. Exchange, the simple diffusion means whenever there is a cell membrane is made up of lipid, right? Lipid. So whenever there is a lipid soluble substance that is coming across the cell membrane, it will transport it. There will be no problem in that. It will diffuse out simply, right? There will be no utilization of any type of protein in that, right? But whenever there is a water soluble substance, which will try to diffuse via this, then it can't. Water soluble substances are diffused with the help of proteins. There will be needing of proteins. Okay, so you can write in simple diffusion another, you can say function, right? Discussing the functions. Exchange of gases in one function, exchange, and another is transport of lipid soluble substances. Many drugs are lipid soluble, alcohol is lipid soluble, okay, many things are there. Then simple diffusion is exchange of gases and transport of lipid soluble substances. So there is a law governing the simple diffusion. There is a law governing simple diffusion, and this law is called the Fick's law of diffusion. Fick's law of diffusion. Fick's law of diffusion has a question in your pre-test. If you are following the USMLE pattern questions, then we have a book called a pre-test. You can just see the book. Pre-test physiology. Pre-test physiology. Which edition recent one I don't remember. But pre-test physiology is a very useful book for your USMLE physiology preparation. It will make your concepts rock hard in physiology. Okay, it's not a theory book. It's basic about questions. Okay, but there are explanations are very well given, right? So you can try solving questions from pre-test physiology. They're excellent questions. Okay? If you want, if you are more interested in physio. Otherwise, you can just leave it. No problem. Law governing simple diffusion is Fick's law of diffusion. The first question in your pre-test, and the Fick's law of diffusion is asked. Okay, which edition I don't remember. Maybe newer editions, other question will be there. Right. So the net transport. Let's write the net transport. You can use any symbol. I'm using J. Is the net transport. Net transport J is equal to minus of A D, minus of A D delta C by delta X. What is this? What is this? A D delta C by delta X. So here in this Fick's law of diffusion, I am writing, these are a bit advanced concepts. So I am covering this concept because it is very important here. As we are talking about simple diffusion in detail, so it is important for us to understand this Fick's law and what are the implications of Fick's law, right? So A is the surface area. This G is the net transport. Okay, there is a diffusion, and delta C is the concentration gradient. Concentration gradient, right? More surface area, more diffusion. You can understand evaporation. More surface area, more evaporation, right? You can understand like that. Or you can see concentration gradient. More concentration gradient means more water will be flowing in osmosis, right? We'll understand. Then delta X is the thickness of the membrane. This delta X is the basically it's the length gradient, or you can say distance gradient, or diffusion distance. You may call it anything. It's basically the thickness of the membrane. thickness of the membrane. And D here is important. D is the diffusion coefficient. Okay, so you understood the Fick's law, right? So diffusion coefficient is basically, and why wrote it in other color, it is unique to every molecule. Okay, diffusion coefficient is directly proportional to the lipid solubility of the substance and inversely proportional to the molecular size. Okay, or you can say square root of molecular weight. So I'll be telling you. So diffusion coefficient is unique to every molecule. It is unique to every molecule, first thing. Second point is that diffusion coefficient is directly proportional. D is directly proportional to the lipid solubility of substance. Directly proportional to the lipid solubility of the substance. It is inversely proportional to the molecular weight. Smaller molecules diffuse faster, right? Molecular weight, right? And another thing is diffusion coefficient, and it's inversely proportional to the molecular. Okay, I wrote it wrong. Just a minute. You can write here, if it is the molecular size basically. You can understand it like that. Square root of molecular weight. You can write this. Okay, square root of molecular weight. It will be easy. It is the molecule. It is inversely proportional to the molecular diameter or size. So molecular diameter means you will, if you cancel the moles on the both side, we're not going to calculation. It is directly proportional to the molecular weight, right? So this is the fact, and based on this, you can calculate the J itself, right? So J is proportional to D, right? J is proportional to D. Am I correct? Yes. So this J will be directly proportional to lipid solubility. That means lipid soluble substances diffuse faster. Important line, right? Another thing is that J is will be inversely proportional to yeah, surface area is there. Surface area will be there, and it will be inversely proportional to which pack? It will be inversely proportional to your delta x itself. Okay, it will be inversely proportional to your molecular weight. So you may derive the intuitions as follows, right? Yeah. So there is a clinical correlation here. If I want to tell you, let's take the case of emphysema. There's a clinical correlation. I'm writing CC. If I write a clinical correlation, I will mention CC in that. Emphysema. Clinical correlations are very, very important. Just listen to me very carefully. So emphysema is, is the smoker's disease seen in chronic smokers. It is one of the disease in COPD. In COPD, we have asthma. In COPD, we have asthma, bronchitis, and your emphysema. Okay. So it is one of the disorder in COPD. What happens is that due to this smoke particles, okay, the tobacco smoke and all, the alveolar walls are broken down. There is a breakdown of alveolar walls, and if the alveolar walls are broken down, then what happens is that if the alveolar walls are broken down, then the surface area available for the gaseous exchange or diffusion reduces. There is a reduction in surface area. Important line. For diffusion, what it means is that if you have the alveoli, a good alveoli, there's the alveolar sac and the one is called the alveoli, right? In case of emphysema, what happens is that there is a corrosion. The walls will be broken down like this. Okay, so this is the case in emphysema. And if it happens, then there is a decrease in surface area for diffusion, right? So A will decrease. A decrease means, you know the formula, right? D A, then what you had? D A, here, what you had? Your thickness, right? By delta X, concentration gradient by delta X, right? So J was directly proportional to A. That implies J will reduce. Net transport will reduce. There will be breathing difficulties, right? Now the diffusion J will reduce. The diffusion capacity will reduce, right? This is one correlation, emphysema. In case of another clinical correlation, let's take another clinical correlation. This will read in respiratory physiology as well. In case of fibrosis in lungs. Let's say there is a lung fibrosis. Lung fibrosis means there is scarring, the tissue scarring of the lungs, okay, due to which the lungs will become this, there will be fibrosed. Okay, fibrosis means there is a more deposition of the extracellular matrix, that will be collagen fibers and all. So there will be consolidation of lung parenchyma, fibrosis in lungs, or you can say pulmonary fibrosis. So what will happen in case of pulmonary fibrosis is that, in case of pulmonary fibrosis, there will be an increase in thickness of the respiratory membrane, right? You can understand this whole membrane will thicken, right? The thickness will increase. There is a increased thickness of respiratory membrane. And if there is an increased thickness of respiratory membrane, what will occur? Your delta X will increase. That implies J is directly proportional to delta X, inversely proportional to delta X. So J will decrease. Again, there is a decrease in the diffusion, right? So this is these are the two clinical correlations you must know from this part. And here we come to the end of the simple diffusion. Let's talk about, let me leave that. Okay. Let's talk about the second thing, that is your facilitated, sorry, diffusion, facilitated diffusion. In case of a facilitated diffusion is a basic kind of diffusion. Some silent points are there, and we have two properties to study in facilitated diffusion. One is called as stereospecificity, another is called as a saturation. We'll study two characteristics which are there in facilitated diffusion. Basic things: no need of ATP is there because it is another passive mode of transport. No need of ATP. There is a need of carrier proteins. Carrier proteins are well needed. No need of ATP, there is a need of carrier protein. So there is a carrier mediated transport. You can say, you can write it as a carrier mediated. Carrier mediated transport, right? Carrier mediated transport. So here we have two important properties to study. One is called as your stereospecificity. Right? Two important properties. Stereospecificity, and another property is called as a saturation. So in simple diffusion, there was no saturation. I'll tell you what is saturation. We'll discuss a graph also, because this graph may be extrapolated in an exam and will be clueless what graph is that, right? So stereospecificity. What is that? Every carrier protein, just listen to my words very carefully. Every carrier protein will be specific for one type of substrate. Stereospecificity. Okay, the structure is specific to each substrate. So every carrier protein is specific to each kind of substrate. Understood? Every carrier molecule is specific to each kind of substrate, right? So let's take the example of GLUT5. So GLUT5, I'm giving one example. GLUT5, glucose transporter 5, where GLUT1, 2, 3, 4, and all. GLUT5 is specific for fructose. It will not bind to any other thing. Okay, it is specific for fructose. Understood? This is about stereospecificity. Talking about saturation. What is saturation? So basically what is occurring is that there is a phenomenon which is called saturation. So these carrier proteins have a tendency to reach their maximum rate of transport. Okay, they have a tendency to reach their maximum rate of transport, after which they will tend to form a plateau. The rate of transport will remain constant. Okay, if if you go on increasing the concentration gradient, at some point there will be a constancy in the rate of transport. Try to correlate what I am telling you, right? So saturation means there is a tendency of transport, you can say carrier proteins, right? Transport will be a bit confusing for you. Carrier proteins to reach a maximum rate of transport after some point. After some point. Okay. After which there will be, after they reach T max. T max is the rate of transport max. The max rate of transport. There will be no increase in rate at whatever concentration gradient. You go on increasing the concentration gradient and other factors, they will not increase at all, right? So this is the basic thing which is there in your saturation kinetics. So I was talking about a graph. Just plot the graph with me. Here you take the concentration gradient. Sorry, don't take the concentration gradient, you will get confused. Here you take the maximum rate of transport. Okay, you can just take rate of rate of uptake. Okay, in the y-axis, and here you take the concentration gradient in the x-axis. In case of simple diffusion, the rate of uptake will increase with the concentration gradient. Okay, like straight line curve is there, right? This is for simple diffusion. But for facilitated mode of diffusion, okay, this will occur. This is the maximum rate of uptake. Let's say where we have reached the T max, the maximum rate of uptake. This is for facilitated diffusion. So you go on increasing the concentration gradient. At some point, it goes on increasing, but after some point, when there is saturation, it remains constant. It remains constant here. And this constant phase is called the plateau phase. It's a common term, right? It's a broad term. Whatever may be the concentration gradient, it will remain the same. Did you understand the graph? Did you understand the concept? Yeah. Then we have some examples of facilitated transport, right? Let's discuss the examples of facilitated diffusion. So one example, fructose. About fructose, I told you. Okay, GLUT you can write. The glucose transporters are all the examples of your Facilitated diffusion, you can just make a mnemonic here. You know about stop codons, right? Molecular biology, stop codons. It's a silly mnemonic, I know, but it may help you to remember. Facilitated stop codons. You can just remember like this. UAG is the stop codon. UGA, UAG. Yeah. So UAG. U means there is a urea transport. A means amino acid transport. And G is the glut. Done. Then we have another thing which is called as the non-ionic diffusion. Okay. Remember we talked about non-ionic diffusion here. In the classification we told non-ionic diffusion. Let's discuss about non-ionic diffusion. Right. I hope you are not getting bored. A little bit theoretical chapter is that I can't do anything at all. Right. But if you are writing notes along with me, then just hold your color pens and colors and just try to make colorful notes. So we'll love the notes forever. Like the basic thing, the basic secret of scoring a decent mark in physiology in any subject you can take. Biochemistry is the dry subject, I also know. Okay. Physiology, just draw diagrams and color. Okay, you color the paper in the shades of rainbow, and the examiner is impressed. Okay, the kid knows everything, and the paper looks appealing to the examiner. Right? They crave for that color pens and highlighters. So why you are not using that? Right? So some universities don't allow, but if it is allowed, then use it to the fullest. Right? Yeah. So let's talk about non-ionic diffusion now. Non-ionic diffusion means I'm giving an example. In the collecting duct, the ammonia that is secreted by the cell into the lumen binds with the H plus ion to form NH4 plus ion. You know this? NH4 plus ion. After diffusion into the lumen, it gets ionized. And this ionization of the ammonia is called the non-ionic diffusion. Okay. So if the ammonia, the NH3, okay, gets secreted by from the PCT, okay, it gets secreted by simple diffusion. But if ammonia is secreted by the DCT, if ammonia is secreted by the sorry, not the DCT, collecting duct, then it is secreted by non-ionic diffusion. Non-ionic diffusion means some ionic substance is converted into non-ionic form and is diffused. Diffusion occurs. This is the basic concept. So what I told is that you have a nephron like this, loop of Henle and all, and here we have the collecting duct. Right, this is the collecting duct, and this is the PCT. You know this. Here you have the host of capillaries, efferent arteriole, tuft of capillaries, the glomerulus, the Bowman's capsule. Okay, the PCT and all, the loop of Henle. If ammonia is secreted, it is secreted in NH3 form. This occurs by simple diffusion. By simple diffusion. But if this NH3 is secreted, the cells of DCT, okay, in the sorry, I'm telling DCT by default. What is happening, I don't know. Okay, no problem. So collecting duct will have cells like this. Understood? Will have cells. Every structure is formed of cells, right? And this must be the lumen. So if I'm zooming this out, if I'm zooming this picture, it will be like this, somewhat like this. The cells will be there. You can draw this diagram to explain non-ionic diffusion in your exam, and the examiner will be impressed as hell, right? Uh, Yeah, this is the lumen. Here what happens is that first thing, this NH3 within the cell, this NH3 will combine with H plus ion, form NH4 plus. This NH4 plus moves into lumen, and this process is called as a non-ionic diffusion. Right? This is the whole point. What I am trying to explain. Right? So we have about you can remember other two examples of non-ionic diffusion if you want to remember. It's not much important. If you want to classify, you can classify. It's well and good. It will fetch you more marks if the examiner is impressed. Right? So basically, you can remember these examples. There is secretion, absorption of weak acids in the stomach. Like in some drugs are there, sodium salicylate and all. You might have heard that drug. If you haven't, just leave it. Salicylates. Salicylates are absorbed in the stomach by this process. Okay. So salicylate is basically weak acidic drug, weak acid, and this is absorbed into the stomach by this non-ionic diffusion. Okay, and absorption of bile acid in the distal ileum. So second example you can write absorption of bile acid into distal ileum. Okay, so this completes our passive mode of transport. Okay, let's move on to the active transport. We have a bulk transport as well. If time permits, we'll discuss that also, and if not, we'll discuss in next class as usual, because I have to have dinner also, right? So active transport, I guess it will take time. No problem. Primary active and secondary active transport, you know this, okay? So primary active transport, let's first come into primary active transport. Primary active transport. Primary active transport, what are the examples of primary active transport? So there are at least four examples which are there of primary active transport. One is the most important example. Of course, you might have guessed. Examples are sodium, potassium, ATPase pump. You must have read the mnemonic Nokia. Sodium out, potassium in at the expense of ATP, right? You must have read this thing. It will allow, just let me write in another color. I like to draw the ATPs in red. I don't know why, but it's what it is. 3 sodium in, 2 potassium out at the expense of ATP. ATP is hydrolyzed to ADP and PI, organic phosphate, right? This is the sodium potassium ATPase pump, right? And if you know, there is a parietal cell in stomach, parietal cell which secretes the HCl, and the formation of HCl, there is a pump. We'll read in your gastric physiology or GIT that there is a pump in your parietal cell that is called the K plus H plus pump, or a H plus K plus pump, or you can write or H plus K plus ATPase pump. Yeah. So it's like Vishwanath Tripathi and Vishwanath are the same and all the same, right? Yeah. So this pump is also known as, there's other name. It's called the proton pump, because H plus is involved. H plus is called proton, right? And in collecting duct, in your collecting duct, there is another thing. Collecting duct has another K plus H plus ATPase with this proton pump is also present here. Okay, proton pump are also present in the collecting duct as well. Another important thing is the Sarka pump. Smooth endoplasmic reticulum calcium ATPase pump. Okay, in muscle cells. So we'll read about Sarka pumps when we cover nerve muscle physiology in muscles. Okay, in muscle cells or muscles. This much is important, right? And yeah, here in primary active transport, we have to read about the sodium potassium ATPase pump in detail, because sodium potassium ATPase pump itself is a short note question. It may come in combination with active transport as a long question. Okay, so the sodium potassium ATPase pump is very important. The structure, the organization of Na plus K plus pump is very, very important. So let's discuss the sodium potassium ATPase pump now. It's a very important 5 marks question. Okay, so here we define a statement which is a term which is called the coupling ratio. Coupling ratio means how much the two ions are getting in or getting out, or diffusion ratio. Okay, that is the number of, here in this case, sodium moving in by the number of potassium ions moving out. So here the coupling ratio will be 3 by 2 equal to 3 is to 2. Understood? Another thing is that it is a ATPase linked pump. ATPase linked pump. That is, for the production of energy, this ATP is being hydrolyzed to ADP plus PI. You know the statement. You can write the statement as well. Okay. And another thing, it is a electrogenic pump. Electrogenic pump means it causes the depolarization of the cell. Okay. The cell will become negative with respect to outside. The cell will become negative with respect to outside. Okay. this thing is there. Okay. Electrogenic pump. It is a electrogenic pump. Understood? And the sodium potassium pump is basically has a common function, has a very important function, you can say, in maintaining the resting membrane potential. Na plus K plus pump. This is called the RMP of the cell, resting membrane potential. The potential of the cell when there is a no impulse conduction is called as the resting membrane potential. Okay, but the contribution is very less. Like if the resting membrane potential in majority of the cell is minus 90 millivolt, the contribution is only 4 millivolt. Contribution is only, you can write minus 4 millivolt. We'll read in our muscle physiology, depolarization, all. We'll discuss that. We'll get complicated here. Right? That's why I'm not telling you. Another function. It is helpful helping in cell volume regulation. What is this new concept? As it involves the exchange of ions, okay, since it causes a net loss of one osmotically active particle, it will cause the cell volume regulation. What I'm trying to say is that, just look at this. This is a cell. This is your Na plus K plus pump. Okay. We'll discuss the structure in a bit. Till then, we'll draw our simple circular structure, right? 3 sodium will be entering. That is, three things are coming in. 2 positive charges are leaving out. So there is one positive charge that will be present inside, right? So there is a net loss of one osmotically active particle. What I'm telling to say is that one net loss of osmotically active particle, that is, the inside of cell will be more osmolarity and the outside will be less osmolarity. Water will flow from less osmolarity to more osmolarity. You know this thing? Or if you don't know, let me tell you. You must have read these things in your previous classes. That's why I didn't feel. So you must have read this. There's a compartment A and B here. Water is present in both compartments, but here the solute molecules are more. More solute means more osmolarity. More osmolarity. Here the solute particles are less. So water always moves from lower to higher osmolarity. Water moves from lower to higher osmolarity. Understood? So water will move from here to here. So it is helping in, so cell will swell up. So it is helping in the regulation of cell volume. Okay. And another thing, another important function is it is in the regulation of BMR, that is a basal metabolic rate. Regulation of basal metabolic rate. Understood? Did you get all the things which I told? Right? Okay. Uh, it's already time. So we will discuss the last thing, and the secondary active transport will cover in other lecture, the second lecture. Okay, because it is sequence of lectures, so no need to worry. If I am leaving a topic in mid, it's fine. Okay, it's fine. Right. You don't need to worry. We'll study about that secondary active transport. We'll discuss it in detail. I promise you, we'll discuss each and everything in detail. Let's say we are discussing in detail. You might have observing that we are not leaving any point, discussing the concepts in very detail, great detail. So we'll cover it in detail. Just we need time. So I can't extend the lecture to 1 hour and 1 hour plus because I'm just having a sore throat and all. No problem, no problem. We'll discuss this concept in next class, but we'll finish this the structure. We'll finish the structure first. Okay. So let's discuss the structure of basic structure of sodium potassium ATPase. If we want to discuss the basic structure of sodium potassium ATPase, so we have a large alpha subunits. It is a trimer. Basically, it is a trimer having alpha, beta, and a gamma subunit. So it's basically, if you want to draw, you can draw. You have to draw an exam. It's not a optional thing. It's a compulsory thing. You must be good in diagrams, right? Let's say it's an extracellular fluid, and here is the cytoplasm. Let me shade the cytoplasm. It's inside the cell, right? It's inside the cell. And here we have a alpha subunit and a beta subunit. Okay, and a gamma subunit itself, and the gamma subunit is very small. Okay, so we have alpha subunit, we have a beta subunit. Just remember this both subunits. We have a beta subunit in the shape of a reverse P. You can just draw it like that because in many books this type of diagram is given. Will be beta subunit, and here the beta subunit is there, and then we have the alpha subunit. Alpha subunit, you can draw it like that. You can draw it like this, having a cleft here for the binding of that sodium, right? So here in this cleft will be binding of 3 sodium, and in this part we'll be having binding of 2 potassium, right? 3 sodium and 2 potassium. Understood? So this is the basic thing about alpha subunit and beta subunit. This is the ECF and the basic structure. This is alpha. Can mark this. If you want to make your notes colorful, you can color these parts. I just like coloring the things up. So if I'm wasting your time, you can save me. Okay. Just shoe type of structure. This is the basic structure. Okay. I'm not going into that shear molecular weight and all details. Okay. It's a basic structure. It's a trimer. Just remember alpha subunit is larger. This is a larger subunit. Yeah, alpha is the largest subunit. And if you want, I will teach you this much is enough. I guess this much content is enough for a 5 mark question, short note, right? So that's all. And yeah, many hormones increase or decrease the expression of Na plus K plus ATPase. This part is important. So hormones increase or decrease expression of sodium, potassium, ATPase also. And what are the hormones? Let me state for you. First one is the thyroid hormone. Okay, the thyroxine. The thyroid hormone will increase the number of sodium potassium ATPase. Will increase the number but not the activity of sodium potassium ATPase. Thyroxine. First one, just right. Will increase number. No effect on activity. Nil effect on activity. Nil effect on activity. Another thing is a ANP. ANP is atrial natriuretic peptide. This atrial natriuretic peptide have no effect on number, but it decreases the activity. Decreases activity, right? Third important hormone is your insulin. What will insulin do? Insulin have no effect on the number itself, but it increases. But it increases the activity. Increases the activity of sodium potassium ATP. Okay, that's why if it increases the activity, that means it is promoting the potassium efflux. If it increasing the activity, it promoting potassium efflux. It is promoting the potassium efflux. That is, it is reducing. Uh, that's why the treatment of hyperkalemia, increased potassium in blood, is insulin. That's why it is insulin plus a dextrose even treatment of hyperkalemia. Insulin is given in the treatment of hyperkalemia. Important. So insulin is very very important as per this is concerned. Okay, and another hormones are there like your aldosterone. Okay, aldosterone. Aldosterone means all. You can just remember like this. All means all effects like increases number and activity both. Okay, both. Increase the number as well as the activity of this whole thing. Next we have lastly we have epinephrine. Epinephrine, norepinephrine have like common effects. So it has no activity, number, no effect on number, but it increases the activity, increases activity. So you can just add this additional point. in your sodium potassium ATP short note. If you want to write more, you can just add this hormones list of hormones, 5 hormones. Otherwise, it is not much important. These things you will encounter in many chapters itself. So I have wrote it in a this format, right? And in next class, we'll talk about the secondary active transport. So next class, we'll talk about secondary active transport and onwards, right? So thank you all. I hope you liked the lecture, and if any doubts remain, you can you must be knowing my Instagram ID. That is Vishwanath.Tripathi underscore. You can text me the doubts there, or you can take my number 9861 999 541. You can WhatsApp me your doubts if possible. Okay, if there is doubt, then you can feel free to contact me. That's all in today's lecture. Thank you very much. I hope you enjoyed the lecture. Let's meet in next class. Till then, bye bye and good night.